The first speaker is a Canadian molecular virologist and clinical epidemiologist with over 20 years of experience in infectious diseases, diagnostics, and surveillance. He's earned his PhD in virology from Griffith University in Australia. He gained international attention in 2023 for his research with DNA contamination in Pfizer and Moderna mRNA COVID-19 vaccines, pointing to high levels of plasmid DNA in the presence of an SV40 promoter enhancer in Pfizer's vaccine. Please give a warm welcome to Dr. David Speicher.
Thank you, Calgary. It's time to wake up the lions throughout Canada. Wow, my nerves are now gone. Thank you, Eric and Daryl. I'm like in the backstage, headphones on, nervous, and I'm like, now I'm fine. All righty, turn her on. Wow. So we've all been told by our government's Health Canada these things. The vaccines are safe and effective. The vaccines have saved thousands of lives. They remain at the site of injection. It just contains mRNA, no DNA, no SV40. It's the genetic material won't enter the nucleus. It's simple, not bioactive, and they don't cause increased cancer. Well, if we look at an early interview with Dr. Dina Hinshaw and Dr. Rillo, they were asked about the whole biodistribution thing at a town hall, and responded, when the mRNA, along with all other intermuscular vaccines, are injected into the arm, the vaccine and the cells remain at the site of injection. There is no evidence, and we would not expect that the vaccine or itself or any of the components would make it into the breast milk. Let's look at what the science actually says. I'm a scientist, I'm about facts. So, up here, we know in mouse studies, the mRNA vaccines will cross the placenta within 1 hour and be found in the fetal circulation, and it could accumulate in fetal tissues, and it has been found in milk. Now, I have personally examined 44 vials of the vaccine, 32 in Canada. This is the data. And yes, in our vials, PCR-wise, the loads are below the 10 nanograms per dose cutoff. This is all fragmented DNA. It's all inside the lipid nanoparticles. And to all of the early vials of Pfizer have significantly increased adverse events. It could be because of the buffer or something else. Now, all of the guidelines for the vaccines are for naked DNA vaccines, not for DNA within lipid nanoparticles. We know lipid nanoparticles inject mRNA and DNA directly into the cells. The size for fragments should be less than 200 base pairs. And one thing of this because of time, I'll keep things short. FDA and the guidelines in 1986 were 10 picograms per dose of plasmid DNA. In 1996, levels were increased to 10 nanograms per dose, and in 2003, WHO gave plasmid DNA privilege status so that the acceptable levels could be leaned in. So at the 1996 levels, which are here, we can see all of ours are above the historical level. Now, we know from 1999 David Dean's research, if you take 3 to 10 DNA fragments injected, transfected into a cell via a lipid nanoparticle, it's just a DNA fragment with a 72 base pair enhancer on it, that piece of DNA will move in a non-dividing cell with an intact nuclear envelope, will move into the nucleus and could potentially, has been shown to insert itself into the human genome. It's been known since 1999, and it has been used against us. Now, how many here have heard about what's going on in Australia? It's been big. I looked at 3 of their vials. All of them, PCR-wise, is way over the 10 nanograms per dose. And based on thermometry, it's even higher. And this here caused or led to Adrian McRae in Port Hedland calling for a council meeting to vote and support this work, and said every councillor must note my report on those vials. They're all above the TGA limits, and this needs to be looked at. TGA came back October and then said it's nothing but misinformation, small sample size, not a accredited lab. And we, a bunch of us, wrote back and addressed all of their allegations and false and misleading claims. And we found, too, that the TGA staff acknowledged that DNA integration into the genome is possible. The TGA knows that the SVV40 enhancer promoter in the Pfizer vaccine can drag DNA into the nucleus of the cells, and it was not disclosed to any of the regulators. The TGA's response was a 1-page. We gave 14 pages of facts back. So, where's their facts? I would like to see them, because I haven't. Now, what about the brand new boosters? Well, last fall, an Irish doctor sent me 6 vials from Ireland, including the brand new JN1 booster. Has it been cleaned up? Is it any better? Pfizer has been pushed on this thing of DNA SV40 for the past 2 years. I tested the vials. It's a plug-and-play plasmid system, which is a round DNA ring, and the part you want in, you shove in. You don't want it, change it to something else. SV40 is still there. The DNA is still way above the limits. This is the 10 nanogram. On the left is a linear scale for visualization. On the right is in log, which most scientists would use. This is the historical DNA from the 1980s. This is the current one. This is all for naked DNA, not DNA in a lipid nanoparticle, and it's all above. I looked at as well 2 new Canadian vials I picked up. They're just as bad across the whole board. Now, so if we compare the Irish versus the Canadian, Australians are still up. It's there. Now, red is spike, ori is blue, green is the SV40. It's 3 parts on the plasmid. Depending on which part you test for, you get a different response based on the way that plasmid was digested. So sometimes spikes higher or he's down, SV40 is up. Still there, still bad. It's not been changed, and these things should be pulled off the market. DNA in the shots is not the only harmful thing. The LNPs and the spike are both toxic. There is N1 methyl pseudouridine in there. It's a plastic-like RNA. It's also cancer causing it causes ribosomal frame shifting, which means the spike itself is not always full intact in there. Sometimes it's short pieces, half, sometimes it's full length, and that makes a off-target immune response, which none of us really want. There's lipid adducts, there's double-stranded RNA, DNA, RNA, hybrids. The more ways we look at these vaccines, the more ways we find in which these can cause harm. Now, well, what about if this is passed on to our kids? I am and my team just getting ethics approval to compare sperm cells in both vaxxed and unvaxxed to see, is this being passed on to our offspring or is it not? If it is, and it's in a sperm cell, if that cell makes an egg, well, meets a egg, makes a offspring, every cell in that body has the code to make spike. Will it? We don't know. I don't think in our lifetime we'll see the full extent of the harms caused by these vaccines. Therefore, as you've all known, seen the Davidson report I and Dr. Dole were a co-author on this, and so it called for a government investigation as to what occurred in the pandemic, what info did the government know. We need a return to evidence-based science, evidence-based healthcare, and not narrative pushing healthcare. We need as well a government inquiry with subpoena powers. We need PCR testing only on people with symptoms and not driving a case-demic. And open information, open informed consent. Our mainstream media has not argued against us with facts. I've heard nothing of facts from Timothy Caulfield, AMA, nothing. All they come up with are cartoons like this that shows us as a monster. And as I say, if they only have ad hominem attacks, you have to own it. I love to fish, and I guess that's me. So you just have to smile and laugh and roll with the punches. Now, on a more serious note, this is what our government agencies have told us. And one thing is for certain, we know they lied. Three weeks ago, Australia, based on my work on the vials and a multifaceted David and Goliath fight, Australia launched the David Declaration, which calls for a return to evidence-based medical science, a moratorium on the mRNA vaccines, and support for all those who are vaccine injured. Following Australia's steps, the North Group has sent my research and concern on the DNA and the SV40 as of last week to 22 nations, the heads of the governments, and Canada is on the list, and that includes Dr. Mark Trozzi and myself. Last month as well, Kevin McKernan, Jessica Rose, myself, Maria Gucci, following Katie Ashby Coppens and Julian Gillespie filed a citizens petition to the FDA, and so far it's got 268 people who have wrote in against this and want mRNA banned in the States and worldwide. So, what are we doing here in Canada? Well, this week, Canadian Citizens Care Alliance is launching in the next 48 hours this letter and is calling on our government of Canada and all of the provinces and medical health officers for a ban on mRNA technology and a moratorium on the COVID vaccines. The tides are turning and the evidence is piling up and they can't ignore it anymore. So, in summary, much of what we have been told is not real. We must follow evidence-based science. We must call for a stop to these shots, a halt on things like gain-of-function research, mRNA technology, and definitely no more gene therapy forced upon people without proper informed consent. And thus, there's so many vaccine injured out there. All of them need our support. They need to be recognized. And I've talked with Maxime Bernier, and I am running in Hamilton Centre as a PPC candidate, and we will make this a federal election issue in Canada. Last point. So, in close, I love this verse. Be strong and courageous. Do not be afraid. Do not be discouraged. For the Lord your God is with you. And I encourage all of you, if you've lost your job for not getting vaxxed, if you are vaccine injured at all, stand up. You have a voice. You have a right. And your story needs to be told. And if that is told and science is followed, we as a society and humanity can start to heal. Thank you, Calgary.
Dr. David Speicher.